Yes, hello everyone. I'll just get started now, and I guess people showing up later will be able to catch up. So welcome to our very first remote Café Sci talk. I'm David, one of the committee members of Cornwall Science Community. I'll be in charge of the, or hopefully well in charge of the tech for this meeting. This is our first time doing it, so if there are any issues, please do bear with us. Just to let you know, we will be recording this meeting and hopefully publish it on YouTube in some sort of form. So if you don't want to be seen, feel free to turn your microphone, your video off, or even change your name. Otherwise, I'll pass you straight on to Robin, who some of you might already know as the chair of our more traditional Café Sci talks.
Okay, thank you, David. Right, well, yes, this is quite an exciting and, that is to say, quite a scary moment as we experiment with using Zoom. Now, I'm thinking that an awful lot of people are getting very, very familiar with Zoom and have done over the last few weeks or so, even if you weren't using it before. But there will be ways in which we're going to have to discover and work out what works best. It's going to be a learning curve evening, in other words. So if some things don't work terribly well, hopefully the software is going to work okay, but in terms of how the process works, we're going to have to be, it's going to have to be, as I say, a learning curve. Traditionally, a Café Sci evening will start with the host, which has tended to be lately me, in Falmouth at least. And I will then introduce the speaker, having first explained the way that the Café Sci program works. We start out with a relatively short talk from our invited speaker. We suggest about 20 minutes or so. There's then a bit of a break whilst you can digest what you've been hearing, and then a question and answer session that can last half an hour, hour and a half or so evening, basically until chucking out time, or when people have to get the last bus home or whatever. So because the Zoom software allows us to mute people when they first arrive, what we'll do is have everybody muted for the first 20 minutes or so whilst Meaghan presents her material. We'll then have people unmuted individually so you can ask your own individual questions. If we unmute everybody, you'll get all kinds of background noise, the feedback's going to go crazy, and so on. So if you have a question to ask, please, unless it's a very specific technical, I didn't understand that acronym kind of question, in which case probably best that you throw that in. Otherwise, please leave your questions until after Meaghan's first presentation, and then there's ways in which you can put up your hand if you have a question to ask. But it's also worth pointing out that at the bottom of your screen, some screens I know have this somewhere different, but there should be something called the chat screen. And if you have a point you want to make or a question you want to ask, and you're not yet able to appear on screen, either because we have you muted, or because you'd rather not, you can always post a question or make a comment in the chat screen, even whilst Meaghan is talking. But we won't let you in to interrupt her until after she's had a fair chance to lay out her stall. So I'm not going to ask if that makes sense to everybody, because we'll then get an enormous cacophony. We're going to sort of Phil Spector-like wall of sound at that point, I hope. Let us go straight on to Meaghan. And Meaghan, you're going to share your screen, I believe, so that you can then present slides and any other information that you have. Meaghan, over to you.
Thank you very much, Robin. Thank you for that absolutely lovely introduction. So today I'm going to be talking very broadly about my research area, which is on phage therapy. And while this is about viruses, it's nothing really to do with our current situation, and will hopefully be a more hopeful perspective of what viruses are and actually how they could benefit us against a threat which is posed to our current society, rather than something which is a problem. So I'll just go ahead and show my screen in a moment. Yep. So you should be able to see the beginning of my slideshow here, I think. Yeah, so phage, the enemy of our enemy. So when I talk about the enemy, what I'm really talking about here are bacteria. So bacteria, not all of them are bad. There are an abundance of bacteria that we really do need as part of our lives. We live with the bacteria every day, and they naturally live in our bodies as well, which are ones that we desperately need. But there are a subset of bacteria which are particularly problematic to us. Ones like pictured here. So we have 3 images at the top here. This green one, this reddish one, and this spirally one here. These are Pseudomonas aeruginosa, and this is one of the main bacteria that I work with. And this is a fairly nasty opportunistic pathogen which can cause very severe illnesses. And there's also bacteria here which you may be more familiar with, things like E. coli, and also MRSA, or by its more deadly name, MRSA. And usually when you become infected or are at risk of being infected by one of these bacteria, what you ordinarily have is a course of antibiotics. And I'm sure most, if not all of us here, have had some antibiotics in our lifetime. And at their time, antibiotics really were revolutionary and have saved millions upon millions of lives since their discovery. But we can't keep on using antibiotics, and that is mostly because bacteria are becoming increasingly more and more resistant to the antibiotics that we have been using. And what this means is the antibiotics that we have are no longer effective, and the bacteria can simply evolve resistance faster than we can really discover new antibiotics. And this problem is mainly coming from how fast bacteria can evolve, and evolution is really my primary background. So usually when I mention evolution to people, they think about it in these really long time scales, literally millions of years, for something like our own speciation from ape-like ancestors, which did take millions of years, but that was mostly confined by how fast we reproduce. So for us, it takes 20, 30, 40 years to reproduce. But for bacteria, that time frame is reduced to minutes. A bacteria can reproduce in as short as time as 20 minutes. So those generation times are much faster, which means a bacteria can evolve much faster than any organism like us. And this is, for example, one bacterial organism which we use to study evolution, because its evolution is quite visually striking. So, for example, all of the bacteria which we had at the start of our culture all originally looked like this very smooth round colony here. But then after literally just a period of 1 week, these bacteria basically grew and evolved and produced more and more offspring. And in those offspring, there evolved random mutations which resulted in all these different colony subtypes. So you've got these 2 smaller ones here and these 2 larger ones here, and these are all genetically distinct bacteria which have all arisen just over 1 week. So it's perhaps unsurprising that bacteria can evolve resistance so much faster than we can really discover new antibiotics. But what this also means is, if we are going to find something to replace antibiotics, or something which can work alongside antibiotics, it needs to be something which can either overcome or keep up with the speed at which bacteria can evolve. And what that something could be is bacteriophage, or for short, phage. So as I mentioned earlier, these are viruses, but they're viruses which only infect bacteria. So so they pose no particular threat to our own human cells. They can't infect our cells at all in the slightest. They only infect bacteria. And their name quite literally means the bacteriophage. It quite literally translates in Greek to the bacteria devourers, such as their name for how deadly these viruses are to these bacteria. So like a virus, what they are made up of is the essential head-like structure, which contains the genetic material, essentially the instruction manual for how you make one of these viruses, or phage. And what it'll do is essentially inject this genetic material into a bacterial cell and force that bacterial cell to produce more phage. And just to give you an idea of what that looks like, I have found a nice animation online. So wait for that to come up. So imagine this is a freely floating phage particle. And what it's doing is it's searching for its bacterial host. And what it'll do is when it finds its host, it will latch on. It will inject that genetic material into the bacterial cell, which will then be arrested and stopped from doing its normal activities. And it will instead be forced to make more and more of these phage particles, literally thousands upon thousands upon thousands of these viruses. And it will essentially be forced to do that until the bacteria quite literally explodes. So not only is the phage effective at killing the bacteria, but in doing so, it creates more and more copies of itself, which can then go on to infect other bacteria. So it's quite unsurprising that phage have been looked to as a replacement or alternative to antibiotics. And actually, phage was one of the first treatments to be considered as a frontline attack against bacterial infections in the 1900s. And actually, phage was discovered in as early as the 1920s, and it was championed by this fellow Felix de Herelle, who championed the use of phage therapy throughout Europe and also Africa and Russia as well. And it particularly really took off there. And the only reason that it didn't take off in Western countries is because antibiotics were discovered in the subsequent decades, which meant that we kind of went one way on our medicinal pathway in terms of tackling bacterial infections, and other countries went their own way. Although they did use antibiotics as well, they also kept up the use of phage. And it's only now that we are faced with these threatening antibiotic-resistant infections that we are now looking to use phage instead. And these have been creeping over into our own countries as well. So there have been a number of cases in the UK and the US as well, where phage has been used to cure otherwise life-threatening infections. So you can see even here, there's an article in the BBC News where this viral cocktail, which means a mix of phage, nothing to do with the alcohol, has been used to save someone's life. And there's also this case here. So this is a case of a man whose story I've read in a lot more detail, and I've read the scientific articles around it. So this gentleman here became infected with a Pseudomonas aeruginosa infection, one of the bacteria I mentioned at the start. And this infection was essentially on his heart. So it was on the major artery, which was feeding the rest of his body with blood. So this was a really sort of life-threatening infection, which persisted for years without being able to be cleared with antibiotics. And it was only when they started to use phage instead that it was actually successful, and this man's life could be saved. And not only that, but his quality of life could also be recovered as well. So what these stories show us is that phage can be effective in ways and in cases where antibiotics simply cannot. And it's part of what I do, and part of what many scientists around the world are doing, is to try and understand what makes phage so effective. And the first question we really ask is, what happens when bacteria evolve resistance to phage? How is this different to when bacteria evolve resistance to antibiotics? And to try and help you and me communicate this to you, I've done these very crude animations. So ordinarily, when a phage comes to infect a bacterial cell, it'll essentially recognize its host by particular structures along the surface of the bacterial cell, and that'll eventually lead to a successful infection, and the bacterial cell will die. So a quite simple way that a bacteria could evolve resistance is to change the structure of these surface structures. And when it does that, the phage can no longer recognize the bacteria as its host, and as a result, it won't be able to infect, and the bacteria can go on along with its normal day-to-day life. But because the phage are also biological and they also have their own genetic material, they too can evolve, and they can also change the structures of their tail particles in order to be able to re-recognize the host, even if they evolve resistance. So as a result, we get a reversal back to our original starting point, and the bacteria is then killed. So what you then essentially end up locked in are these evolutionary cycles where the bacteria are trying to evolve resistance to the phage, but then the phage basically overcomes that and evolves to stay infective against the bacteria. And because this is a cycle, when we think about it in terms of a phage therapy perspective, there are essentially three main outcomes to this cycle. And the first is that the cycle keeps on going. So we could end up with a circumstance where the bacteria are constantly battling out against the phage, but aren't entirely cleared out. And the phage are constantly evolving to keep the bacteria in check. So what we are left with essentially is a partially phage-resistant infection. But then we have a second outcome where the bacteria can't keep evolving resistance against the phage, and they're essentially wiped out by the phage infecting them. And for a patient, this means that the bacteria are completely cleared from the host. So for outcome number 2, we would consider this a treatment success. And then we have a third outcome, which would seemingly be the least favorable outcome, is where the phage can't keep evolving to reinfect the bacteria. So what we are left with is one outcome where we have treatment success. So the phage has done its job here. And then we have 2 outcomes where we're left with a phage-resistant infection. So the next question we really need to ask before we bring this into our regular medicine is phage resistance all that bad? Is it bad for us being the bearers of the phage-resistant infection? And is it bad? And is it bad for the bacteria as being phage resistant? So to understand this, we need to look closer at a bacterial cell. So as I mentioned earlier, they're essentially blobs with lots of these different surface structures by which the phage recognize them. So a quite strong way that a bacteria could evolve resistance to phage is to simply lose these structures. It could essentially not bother even changing the structure of them, it could just lose them. So for example, if a phage recognized it by these tails, it could lose these tail structures and stop the phage from infecting at all. But in losing those tail structures, the growth potential of those bacteria is really significantly reduced. It can no longer swim about, it can no longer move nearly quite so well. And as a result, its growth and its survival while it's infecting us is really, really reduced. So for us, this could be really beneficial because if that bacteria is infecting us, it's really no longer that good at infecting us, and it could be cleared by other things like our immune system. And another way that bacteria could evolve resistance is to try and cover up these surface structures. So it could produce this gunky material we call biofilm, which essentially builds up these walls between the bacteria and the phage to stop the phage from reaching them and infecting them. But this also has a cost to the bacteria because it has to invest nutrition, it has to invest resources and time into building these structures. So there is also a potential cost for the bacteria there because it's having to invest its resources in defending itself against phage instead of infecting us. And we can even see this defensive structure by our own eyes. So these are cultures from the lab I work in. So on the left here, we have these phage susceptible cultures where the bacteria are just growing happily in the media. You can't really see them all that well, apart from the fact that the media has gone really, really cloudy. But then you have on the right here, you have these phage resistant cultures, and you can really see along the edges of the vials where these bacteria have tried to resist the phage by producing this horrible and gunky defensive biofilm structure. So you can really think of phage therapy as a war between bacteria and phage, where the bacteria are trying desperately to build defenses against the phage, and the phage are trying on an evolutionary level to overcome those defenses. But because the bacteria has to then invest in defending itself against the phage, it's no longer so concerned or can no longer take up so much time and energy in infecting us. And this could be quite beneficial because it could make the bacteria more vulnerable to the antibiotics it was priorly resistant to, and it can make them more vulnerable to things like our immune system, which could then have a fighting chance of actually clearing this infection. So if we want to think about how we could also push this war in the favor of us and in the favor of the phage, we essentially have to push the evolutionary war in favor of the phage so the bacteria can't evolve a complete resistance against so the bacteria can evolve complete resistance against the phage, and the phage could essentially just completely wipe out the infection. Or we could make phage resistance very costly for the bacteria. So even if they can't directly be cleared by the phage during an infection, they could at least be cleared by other things like antibiotics or our immune systems. So that really sort of covers the science of what I wanted to go over. So we can really get into more of a discussion and you can ask the questions that you really want to get into. But like all good science, I don't do any of this on my own. I am part of an amazing research group who have taught me the ways of bacteria and phage. So I would like to pass back over to David and Robin and to you to try and answer your questions. And I will stop sharing my screen.
Okay, and I'm now unmuted, is that right? Thanks, David. Right, well, sometimes I feel my age, you know. You are, a lot of the people that I come across, particularly from the Exeter group, are so skillful in putting over a message, using cartoon characters and interactive stuff and so on. I just think, oh my, this is exciting. I love those kind of cartoony pictures. So that was, in some ways, from my point of view, that was the easy bit. That's where I introduce the speaker and sit back and just let the speaker do their thing. Now we come to the more interesting and much more experimental bit, which is where we try and manage a discussion between a string of different people. We have somebody else just arriving in the waiting room, I see. Let's start with, do we have people already with particular questions that they wanted to ask?
We have one question in the chat here by Cara, who's asked, do I see phage therapy overtaking or replacing antibiotic use? So what I think we're going to end up with is a state where we don't completely stop using antibiotics. We're probably going to keep up using antibiotics for quite simple infections. For example, if we just get an infected graze or an infected cut, we'll probably just keep on using antibiotics. But I think more and more we're going to be using phage alongside antibiotics, because using them together basically creates this double fronted attack against the bacterial infection, which means that the bacteria can't keep trying to evolve resistance against the phage, and it can't keep trying to evolve resistance against the bacteria. So I think what we're going to end up with is a more symbiotic relationship between antibiotic and phage therapeutics. But thank you for your question.
So as I understand it, the phages will either destroy or at least weaken by draining energy, weaken the bacterium, and at that point the antibiotics can then affect. So you've got a pincer movement in military language. Okay, David, am I right in thinking that you're scanning to see what questions we have coming up? Right, I'm not hearing from David.
There we go, sorry for that. If anyone does have a question, please indicate that. You can do that on screen just by giving us a wave into the camera. I'm trying to screen the faces, or on the chat as Cara already did, or there's the possibility of sending reactions. So if you put any sort of thumbs up or applause, I will ping that over to Robin.
Okay, great. So David, I'll leave you scanning and let me know if there are people with questions to ask. In the meantime, whilst we're waiting for this to bubble up, let me ask a question of my own. What are the risks in using, granted that phages will evolve very rapidly, are there risks that they will start to evolve to attack the bacteria that we do want, that are useful or beneficial to us? And how can that risk be managed?
So there is, I guess, close relatives between the bacteria which can be dangerous to us, the pathogenic ones, and the bacteria which naturally live us. So E. coli is a very good example of that, because there are E. coli which naturally live in our own guts, and there are E. coli which cause us quite severe disease. So what we can do is to try and do some preliminary experiments with the phage to see whether or not the phage will essentially evolve to switch hosts. That's one measure we can do. And another thing is to really consider the impact that the phage could have on those populations. So the phage are really very specific to their hosts, even if they evolve to then switch to a different strain or a different bacteria. It's probably not likely to have such a colossal impact on the host microbiota, because it's still even going to be then limited to the populations that it does infect. So even if there is a tiny bit of crossover, it's probably still going to be primarily infecting the pathogenic bacterial population, and only have a very limited or no effect on the other microbiota.
Okay, great, thanks. We've got a couple of questions really around the same subject here. One from Sally. How is it not possible for the virus to mutate to be able to infect a human cell? I think that you just answered that question, but Sally, if you feel that there's a further rider to that you'd want to add, then please do come back again.
I could expand on that. So it's not so much that we need to do preliminary experiments. It's mostly because of how different human cells are to bacterial cells. So those surface structures that the bacterial cell, that the phage will use to recognize a bacterial cell, are not ones or anywhere near close to what we carry on our cells, and our cells are very different in their structure to that of a phage cell. So the leap that a phage would have to do to evolve to infect our own cells is just way too big, and it would actually probably be more beneficial for, like Robin says, for it to evolve to infect a different bacteria than it would to infect something like us. But it's a very good question.
It is. I'm going to splice together, I think, two questions that we have, and that seem to be two halves of the same question. Firstly, when do we think phages, firstly, is antibiotic resistance not something to be concerned about for the future? Or another way of saying that is, when do we think the phages might come to our aid and become part of mainstream medicine?
So in terms of whether it's something we should be concerned about, it's definitely something which we need to prepare for. And the advantage here, the current situation we're in, is that we can see this is something which is coming, and as a result there's already been a huge investment in research to try and tackle this, and there's been a huge investment in research to not only look at alternative treatments, but also in terms of how we can prevent antibiotic resistance from keeping emerging up. So it's something that we need to be continuing to work on, and something that we need to continue to work towards, but until phage therapy and other therapeutics come into mainstream medicine, it is something we need to be at least concerned about, and maintain our momentum in our research. And then to answer your sort of second half of your question about when do we expect it to come into mainstream medicine, it's quite a difficult one to ask, just because of how many logistical hurdles we have to overcome to get something like phage to regular medicine. So phage is quite different from all other treatments which we tend to use in Western medicine. It's something which is alive. So when we think of how resistant some people are to things like vaccines, which take something which is alive to make someone immunized, we can think of how concerned people will be when we're taking someone which is alive to treat an infected person, and that living thing could stay on infecting that bacterial infection if the infection can't be cleared. So you could end up with phage persisting inside the human body, which is something that we need to then answer is, what are the consequences of this? So we're closer than we are 20 years ago, when this started to re-emerge as something of interest. But in terms of timescales, what we really need at the moment are more clinical trials. There have only been really a handful of them so far. So once we really get more concrete clinical trials, it is something that could potentially come into fruition, hopefully, and it should do within the next few decades, or next few years, hopefully. Fingers crossed.
Fingers crossed. We've got a couple more questions probing that issue about what's holding us back. But before we do, we've got from Henry an even more basic question. I love the really basic questions. Where do phages come from? And
I love that question.
and also the other side is, where do they go? What happens to them once all the bacteria have been blasted? I love the language here. Meaghan, where do phages come from, and where could they go?
So I'm really glad you asked that, because it's something I purposely didn't cover, because phage can quite literally come from everywhere. So there are phage wherever there are bacteria. So there are phage naturally living in our own guts, because we are full of bacteria which help us digest our food. And there are phage which naturally infect those bacteria. So there are phage in our soils, there are phage in our carpets, on our skin. They are absolutely everywhere. So even if if, for example, we did get a phage resistant infection, it shouldn't be that hard for us then to find another phage which could infect that bacteria, because they are literally so abundant. They're the most abundant and most diverse living thing in the world. So what this means is, because we also naturally interact with phage on a regular basis as well, it also means that they're likely to be a very safe therapeutic, because there's something we interact with so radically, we shouldn't have an adverse reaction to phage either. So in short, phage everywhere.
And where do they go?
Where do they go? Well, if on like a seasonal thing, where there are bursts and blooms in phage abundance, phage naturally decay in the environment. So like all viruses, they're naturally vulnerable to environmental stresses. So they could be just naturally degraded in the environment. Or phage can also have a very clever tactic. This isn't all phage though, and not the phage that we try and use in treatments, is that some phage can also just integrate themselves into the bacteria. So they can just integrate themselves into the bacterial genome, particularly if the bacteria is quite slowly growing. Then the phage will essentially hitchhike into the bacteria and basically benefit off the bacteria's growth. But those aren't ones that we try and use in treatments, just because we don't want it then to just hitchhike on an infection.
Okay, can we take that basic question back even more globally? Where the phages come from to get into your Petri dish, they come from all around us. But in evolutionary terms, sounds like these things are very, very, very early in the evolution of life. Do we know that? Can we tell that? I mean, presumably they don't leave a fossil record.
No, so there'll be no fossil record of phage, because of how small they are. We literally have to use an electron microscope in order to be able to actually even see what they are. But it's very likely that phage came as early as bacteria has evolved. So it's probably, so in terms of genetic elements and things which could just self-reproduce quite simply, are probably something that's going to be very closely related to phage. So phage is probably one of the earliest kind of life forms which arose in our evolutionary history.
Sounds like it, doesn't it? Okay, we've got a few more questions about the development and what boosts and what constrains the development of phage therapy. But before we move on to them as a cluster, we've got Hugh from our good old Cornwall Science community asking, those three outcomes that you spelt out at the beginning of no effect and so on, is it possible to predict between stalemate bacteria and phage wins? is it possible to predict which is going to happen, or do you just have to let it roll and see?
So there is probably some different hallmarks which we could consider to make each outcome more likely. So I guess the most favourable outcome being complete treatment success and the bacteria being entirely cleared from the patient. That's probably going to be most likely in infections which are fairly localized. So things like an acute infection for like an infected cut, or even something like an ear infection, or even like a toe infection, for example. Something that's fairly localized, even in that patient that I mentioned earlier with the infected heart. The infection was still, even though it was a chronic infection, it was very localized to his heart muscle. But in terms of the other outcomes, something like the cycle keeps on going, or the bacteria evolve very strong resistance to the phage, that's probably going to be more likely in a chronic infection. So one of the most dangerous sort of infections people can get are things that, for particularly for cystic fibrosis in patients, they get a lot of infections in the lungs, and those infections literally can persist for decades. And over that time, the bacteria will naturally evolve anyway and evolve to adapt to the human host. So a bacteria which is already fairly well adapted to the human host and is already very well deep into the tissues is probably not going to be entirely cleared by the phage, but may be altered by the phage in a way which could make it less deadly. So by picking up these sort of hallmarks, we could probably get a better idea of the predictability of that. But something that I'm going to be doing also over my PhD is to try and understand the predictability of these outcomes. So I'm receiving samples from Brussels where they do phage therapy as a normal treatment. And what I'm going to be doing is seeing how the bacteria have evolved in the patient and how the phage evolved as well. And I'm going to be replicating that evolution in the lab as well to see how predictable these two are. Because if we can predict a patient's trajectory from simple laboratory experiments, we could potentially predict their treatment outcomes as well.
So if that's what you're working on in particular, that's a very good question from you there. Just one thing, as I understand it, we have broad spectrum antibiotics. In fact, part of the problem is that because so broad spectrum, we dish them out like Smarties, and that way you get the resistance. But from what you're saying, there are no broad spectrum phages. They are specific to the particular war with a particular bacterium. Is that correct?
Yeah, I mean, phage can vary a little bit in their specificity. There are ones which could infect similar species or species within a genus, which is just a similar group of species, like monkeys, for example. Not that phage infect monkeys, but monkeys belong to one whole group. But there are even phage which just infect specific strains of bacteria. And something else that we can do in the lab as well is we can pre-adapt phage to particular bacteria in the lab to even make them even better at killing that bacteria. And that's something we can not just do with a particular bacterial species, it's something we could do with particular patient isolates. So we could potentially make phage even more deadly against bacteria by just doing some very quick experiments in the lab before we actually use them in therapy as well.
So you're speeding up the process of their evolution, but they're almost encultured.
Yeah, so we just give them a little bit of a head start against the bacteria by letting them know what's coming.
Like an aperitif, yeah. Okay, this is taking us again into some of the issues that people have raised about what's the cost. Is it difficulties with the cost? Is it difficulties with the delivery? Are there vested interests that are holding back the development? So we've got various different questions. Is it due to costing that phage therapy hasn't become mainstream? Is it because it would take time to identify which bacteria are infecting us and then which phages are best suited? And then how much further along are some countries like Russia in developing this, given how much time they've been doing it? And finally, intellectual property. Do companies that are doing this work, or research institutes, do they own and then sell, market their particular treatments?
Okay, so in terms of, is it costings or is it time which is holding us back? No. So phage, because they're self-replicating and because they replicate so quickly, they're actually very low cost to produce. I can naturally get 10 to the 10 to the 10 to the 10 of millions of phage within one day. So from one phage plaque, I can get millions upon millions and millions of these phage. So they're really fast to reproduce, and they're really easy to store as well. So it isn't particularly costs or time which is holding us back. Also with time as well, it's also quite quick to test phage against bacterial strains. So there was a case recently, I say recently, in recent years, where a boy was infected, had a leg infection with a bacteria, and they had to go through and screen that bacterial infection against 150 different phage that they had in their phage library. And that took a couple of weeks, but in terms of trying to find a targeted therapeutic, it was quite relatively quick. The main things are primarily, as I mentioned earlier, the logistical issues of how you treat or how you consider a treatment which is alive. And there are even some like small prints in our current regulations of what's allowed in treatment, which means that phage can't necessarily be used or used as effective. So one of these things in America is they don't really use combined treatments quite so much, or they're not allowed to. So that's particularly problematic for phage therapy, because phage are most effective when they've also got lots of other similar phage in this sort of cocktail mixture, which makes them more effective. So there's quite a lot of legal issues that needs to be overcome, quite a lot of logistical issues, and also quite a lot of collaborations as well. So phage therapy studies so far have been used to, phage therapy studies in the clinical setting just mainly focus on the clinical outcomes, so patient outcomes and all the outcomes of the mouse or whatever's been infected. And as a result, we don't really know what's going on in the bacteria and the phage inside the host. So we lack a mechanistic understanding of what's going on under the hood of phage therapy. And usually for myself, an evolutionary ecologist, we mostly study bacteria and phage in a glass vial. And that's far, far removed from that of a human body. So at the moment, our current separate fields really do need to meet in the middle. And this is partially what my PhD is also going to be doing, is to try and bring these separate spheres together and try and work together with clinical biologists and evolutionary biologists and ecologists as well, to really get an understanding of what phage therapy is. And that kind of also answers, I think another question was, how far ahead are other countries like Russia? Because they've been using it for a longer period of time, but that doesn't mean they've been using it completely effectively. Not to diss their phage therapy so far, but what phage therapy became in the early and mid 1900s was not this really well understood treatment. What they would essentially do is they would essentially purify phage from either from things like stool samples where the phage are growing quite abundantly with pathogenic bacteria, and they would use this as a very undescribed phage mix for treating an infection. So they don't really know what phage are in there. They don't know how abundant they are, and they don't know which ones are targeting the particular bacteria. So it's really kind of a chance treatment, which no real research has been looking at what goes on underneath in terms of the bacteria and the phage inside the human body. So while they've been using it for longer and they have made advances in terms of developing particular therapeutics, there still needs to be a lot more done to understand how we can maximize patient success.
So it was a bit of a scattergun approach then that didn't allow you to fine-tune the learning. And I seem to recall from a previous discussion we had that one of the issues with phage therapy was that in the West, of course, we're all very gung-ho for antibiotics, and that's where all the big pharma money was going. Whereas a lot of the phage research in Russia was being written up in Russian.
Mm.
And
So
it
yeah,
was,
that's also another thing. All the papers in Russia are in Russian. So this initially had a bit of a crossover problem in terms of how you then translate that research over to other countries. And it's only in recent years with technology and the internet that we can start to bring these otherwise separate research fields together. And this is partially true for those same Eastern European papers as well, because I still find papers which are completely in Russian and I have to use Google Translate to translate paragraph by paragraph what they're saying, but it is becoming a little bit easier to find where different countries are in phage therapy research.
and so if this stuff is being done now in the West with perhaps greater precision or whatever, can we just come back to that question? I think you touched on it, but I'm not sure if you fully answered it. Can undue companies in the capitalist world, can they own and sell specific phage treatments? Can you have intellectual property over something which is A, natural, and B, evolving very rapidly?
So there are people who do sell phage. So in the UK, there is a company which sells phage therapeutics, not for human consumption, because that's I don't think that's legal, but they use it for agriculture. So they use it for improving food storage. So they'll use phage which target bacteria which degrade food, and they'll help use it to help preserve food. So there are phage currently on sale in the UK if you wanted to buy some. I wouldn't recommend it because you can go out and get in some from your natural soils anyway. And in Eastern European countries, you can buy phage basically over the counter. So that's how phage therapeutics took off in those countries, is you could essentially get over the counter medicines for, say, if you had a stomach infection or you had having digestive issues, you could take a phage cocktail associated with bacteria which infect your stomach. Or if you had a burn and that became a little bit infected, there is a phage therapeutic called Phage burn, which can use essentially as a cream to go on your skin, and that is marketed phage. So in terms of the marketability, if you have a unique phage mix and you apply that separately than what has been done before, then you can patent it as a phage therapeutic, providing that's something that is legal in that country. So even though phage can change and they can evolve, that's something that's not really sort of considered or taken into account when these are prescribed.
Okay. We've got a couple more questions lined up. If anyone else has others, please do pile in. And another one from Henry with those basic questions again. I'm going to save till later because I think it's going to open up a can of worms or whatever else you've got growing in your soil. Let's first go to Sally, who has another question. This is actually quite close to my heart. A lot of my work is around homelessness, and there's a big concern about drug-resistant TB within the homelessness population. Are there effective phage treatments for multi-drug resistant TB infections? Very specific area, but on the other hand, it's a serious public health concern.
So TB isn't my research specialty, but I have done some reading a little bit around TB, and I haven't seen yet that there's actually been any movement in terms of phage therapeutics for TB. One of the things which might make it challenging for using phage therapy against TB is because of how TB resists antibiotics, which could also confer partial resistance to phage as well, in that when TB grows in lungs, it eventually forms these tubercules where the bacteria lives at the center and then it gets covered in this resistant shell, which prevents anything, including immune cells, including drugs, including anything else from getting in there. So that may be an instance where phage could be a little bit more challenged in terms of treating those kinds of infections. And it may be a case here that if we do try and develop phage against TB, is something that we would have to target very early on in an infection stage. So it's those kinds of infections where phage therapy may still struggle, as do antibiotics, but hopefully it's something that will be looked at in the future.
That's a tricky one then. Okay, now, actually that takes us to the question of Henry's earlier question about where do phages go. But let's go back to Henry's. I think this might be quite a big question. How do phages come into the story of viral infections such as coronavirus?
So with things like coronavirus, it's quite different because we can't use phage against viral infections. Phage are only things which we can use to target bacterial infections. And to think about it more broadly in terms of pandemics and diseases, I don't think, well, it's hard for me to say, but in terms of where phage therapy are going to come in the future, it's going to be more combating this slow rise of antibiotic resistance and antibiotic resistant infections. And it's going to be less concerned with trying to then push back against a sudden viral or bacterial pandemic. So in recent years and over the past sort of 20 and 30, 10, 20, 30, 40 years, it's mostly been viruses which have posed a particular threat in terms of these very sudden and quite scary things. When we think about Ebola, Zika, SARS, and Corona, these are all viral infections, and HIV as well is also a viral infection. So viruses are a lot more challenging to find treatments to, because we then have to develop antiretrovirals and things like that. So in terms of treatments, they're quite dissimilar, but they are targeting the very broad thing, which is emerging threats to human populations.
So phages eat bacteria. Is there anything that eats phages?
So there is so yes, in a way. So phage can also infect other phage at some level. So there are really big phages called jumbo phages, and these are the basic giants of the phage world. And these can even be infected by other phage. And even phage themselves, if we got different phage species almost infecting one bacteria, they could essentially hybridize and mix within an infection, and you could end up with a bit of a mix of the two in the phage progeny as well. So that's really sort of where they start to come in, but they are really sort of they're the basic threshold of what we consider something as being alive. So there isn't really much that goes on between what does or does not infect other phage.
So there's not much good eating in a phage then?
No.
Not very meaty. Okie dokie. At the moment we've got a slight lull. Aha! Henry again.
Sorry to just butt in. I think there was a question earlier from Sam that wasn't picked up yet either.
What's there?
Which was, do you think there might be an issue with resistance developing against phage therapy, just like with antibiotics?
So,
thank you, David.
So the most problematic form of phage resistance that could emerge is if phage resistance emerges without these costs that I mentioned earlier. So without those costs, bacteria could become resistant to phage, but maintain their ability to harm us. And there is one or two studies so far which have shown us some mechanisms, but whereby this might occur. Things like CRISPR, which you may have heard of. Bacteria can evolve resistance by that mechanism, which is less costly for the bacteria for the bacteria than other things as well. But even by these other less costly resistance strategies, if a bacteria, for example, has CRISPR, it can't do things like gain antibiotic resistance quite so easily, because it can't exchange genetic material quite as easily either. So there are certain limitations to different resistance strategies that the bacteria can uptake. And it's part of what we need to look at in research, is try and predict how those resistance strategies may emerge in patients. And then we need to use particular antibiotics, and then use particular phages as well in complementary to try and stop that from happening. So it's something that we do have to consider in phage therapy, but it's something that we may be able to predict in advance and try and overcome.
And then customize to a degree.
Mm.
And back to Henry again, are there harmful phages in the sense of harmful to humans, either because they attack things that are beneficial to us, or they actually attack us in some way?
I guess there's quite a complex answer to that, because as I mentioned earlier, phage can't directly infect our own
You're good at
cells.
those difficult questions, Steve.
They can't directly infect our own cells, so any harm that they do to us has to be very indirect. So it has to be something that they do to bacterial populations that we do need. So, for example, do bacteria decrease or affect abundances of, do phage affect abundances of bacteria, say in our gut, which we otherwise need? Or are there phage in the environment which decrease or increase the abundances of bacteria which are dangerous, or decrease the abundances of bacteria which are not harmful? And to answer that side of the question, is we don't really know, because we don't really understand what's going on between the bacteria and the phage inside of people. We have very little knowledge, or know very little about the phage which infect our own gut environments. So that's very poorly understood at the moment. But that's actually reminded me of some phages. As I mentioned earlier, there are some phages which can integrate themselves into a bacterial cell. So there are some phages called prophage, which can integrate themselves into a bacterial genome. And in doing so, it gives the bacteria its genes as well. So if that phage is carrying a gene which is dangerous, things like a particular toxin or a particular growth factor, then the bacteria can inherit that, and that can make it more dangerous to anything that the bacteria then goes on to infect. But when we design therapeutics, we screen the phages, and this part, the preliminary research is important. We screen the phages to make sure they're not carrying any of those genes which the bacteria could pick up.
So this is where you can see it coming and you can divert it to some extent. Possibly another example, or possibly not an example of that kind of harm. You mentioned one way that bacteria can defend themselves is by setting up that biofilm, the sort of cocoon around them. Could that potentially at some point become a problem in itself if our lungs, say, were clogging up with that kind of cotton woolish substance? Is that the kind of risk that we could imagine?
So that's also a good point. So usually when we think of these very dangerous infections in people, usually these are characterized by biofilms which occur naturally in those bacteria as well, because usually in, say, a chronic infection where the bacteria have adapted to the host, they have naturally evolved biofilms anyway. So if we anticipate a bacteria could evolve resistance by biofilms, we then need to change our strategy to then target biofilm structures. So we can use phages which encode particular enzymes. So these are proteins which basically degrade other molecules. And we can find phage which can basically break down biofilm. So part of phage therapy research is in targeting biofilm related infections. So things like urinary tract infections or catheter infections are characterized by lots of biofilms, and also lung infectious swallow are also characterized by biofilms. But the research on that has been quite promising so far in showing that phage can be quite effective at actually degrading those biofilms and working with other antibiotics as well at breaking those down.
Okay, well, at the moment, the last question we have, unless I can provoke somebody else into coming up with another one, is from Ben, which I think is Ben Holt, or B Holt. Why didn't you call your talk Phage Against the Machine?
Because I clearly asked the wrong people for advice on what to call my talk. But for my next public talk, that will be absolutely my go-to one.
Although I think our enemy's enemy was also pretty smart. Now, Ben, as it happens, is our speaker for next week's cafe. Ben, would you be happy to just come on screen for a moment and then tell us a little bit, just a kind of pre-aperitif about what it is you're going to be wanting to talk about?
Sure, can you hear me?
Yep, we got you.
Looks like I'm completely in shadow. This is let me come up. There we are. Can you see me a bit better? There we are. That's okay.
That's a lot better.
Yeah. Yeah. Hello, everybody. Yeah. So I'll be doing this next week. Yeah, I was quite relaxed about it until I saw that unbelievably professional and fascinating talk by Meaghan. Good work, Meaghan. Yeah, I'll be talking about citizen science and in particular the use of citizen science in the world of marine biology and learning about our marine environment. It's an area that I've been involved with for, I guess, over a decade now, initially purely in terms of researching data that's been collected by citizen scientists. And I've ended up getting more and more involved on the ground. And nowadays, I run a citizen science organization called the Rockpool Project. So I'll be maybe taking us through that life journey that I've been through and explaining a little bit about the opportunities and challenges in that whole world.
Great. So we'll look out for that next week, folks. This time seems to suit a lot of people, but we're going to run it at this time for the next few weeks or so until we see what suits. If anybody has any feedback as to what worked, what didn't work, what you found frustrating in particular, we want to learn from what isn't working in particular. We've got various different email addresses around, and please do contact us and let us know how we can improve this, how we can make this work even better. Just want to make one last little link between phages and next week's talk around citizen science. Meaghan, you were saying that there are these little critters in every patch of soil or probably every glass of water. What prospects are there for ordinary people, ordinary citizens, to experiment a little bit and maybe even share whatever we might learn? Is this something that's really only doable in labs, or can we all explore this interesting, intriguing new world?
I guess to a certain extent it's something you could play around with. The main difficulty is isolating phage, because to do that we use chloroform, which is quite legendarily as that chemical which used to knock people out in spy films. So that's not something which is freely available to the public. It's something we keep close and dear to our hearts in laboratories. So, but if you did, for example, buy phage, then it's something you can play about in cultures in your homes as well. So if you did buy the phage from that UK company to target fruit or vegetable degrading bacteria, and you had a culture of that bacteria, then you are within your own remit to try and grow that bacteria in whatever you see fit and blast it with phage to your heart's content.
Okay, and the link with chloroform incidentally gives us another delightful little link with the talk, not next week, but the week after. Most of you will be aware that Humphrey Davy, the famous early chemist and physicist, was a local lad from down here in Cornwall, from Penzance, hence one of the schools down here named after him. And Frank James, who is in effect the nation's and probably the world's expert on Humphrey Davy, is going to be talking on Humphrey. The link was that Humphrey used to use laughing gas and experimented on himself. So I think he was an early citizen scientist as well as an early scientist. Right, I do believe we've probably come to the end of our, somebody, Graham Greaves saying this time is good. Thank you very much, Graham. I would imagine for those people who are here, it's because this time is good for you. So we'll try and not, we'll try and open it out. Maybe run other events another time, just to see what works. Please do send us some feedback, any other comments and any other suggestions that you have. But for now, well, traditionally at this stage, we have a round of applause for the speaker. So David, could you unmute us all and we'll try a quick round of applause. You can do it visually.
I can try.
That'll have to do.
Very Thank you all very much for your questions. It's a very interesting discussion as well.
Great, great. So folks, hopefully see as many of you as possible back next week. I think the quick takeaway learning is that this is workable. So we'll start to advertise it more broadly. If there are lessons we can learn, please let us know. But my feeling is, yeah, we can do this. So thank you very much, and I'll see as many of you as possible next week.